Welcome back to Beyond 101. Last video, we began our discussion of the innate immune system by discussing our anatomical barriers, and in this video, we're going to continue with the innate immune system by introducing the complement system. The complement system is made of a series of proteins that both propagate and amplify immune responses. And they do this through three different methods, one of which is through what's known as membrane attack. The proteins of the complement system can literally arrange themselves into these what are known as membrane attack complexes and can disrupt pathogenic cell membranes, but that's something we're going to get into in a later video. The second thing they do is they promote inflammation. And the last thing they do is they coat the surfaces of pathogens, and that coating signals for phagocytes, including macrophages and neutrophils, to devour the pathogens. Now, this last process is known as opsonization. And this is going to be the majority of what we focus on in this video. In addition, there are actually three different pathways in which the complement system can be activated. So 3 activation pathways. And these are known as the classical pathway, the alternative pathway, and the lectin pathway. And it's these three pathways that we're going to be focusing on primarily in this video, focusing on how the complement system activates and how it leads to opsonization. Before we get started, though, we need to discuss the nomenclature and conventions used when describing the proteins of the complement system. And unfortunately, these rules aren't always consistent. There are a number of exceptions, but I will make sure to note when we reach those. So the first convention is that the proteins generally follow the form C followed by some number, N. So for example, the proteins of the complement system include C1, C2, C3, and so on. There is an exception, though, and those are going to be the proteins of the alternative pathway. In this pathway, we see some proteins that don't start with the letter C. The second rule is that the proteins of the complement system are similar to what we saw in the last video. They're proenzymes. Complement proteins are proenzymes, meaning that they only get activated when a part of it is cut. Another word we use for this is a zymogen. And it's important to note this because a complement protein, CN, is generally going to get cleaved into CN little a and CN little b. The exception to this rule, again, unfortunately, all these rules are going to have exceptions, are going to be the complement proteins of the classical pathway. The last rule that we're going to focus on is with regards to the fragments of our complement proteins. In most cases, CNA is going to be smaller than CNB. So if I have C3, we know that's going to get cleaved into C3A and C3B. And based off this third rule, C3B is the big fragment, and C3A is the small fragment. And again, unfortunately, we do have an exception to this rule. The exception lies in the protein C2. C2. Again, it still gets cleaved into C2A and C2B. However, C2A is actually bigger than C2B. C2B is the small fragment. And I know that this is a lot, but it's going to make a lot of sense once we start going through the actual complement activation pathways, and you'll see how these rules interact and how we can use them to describe what's going on. The first pathway we're going to discuss is the lectin pathway. And the lectin pathway is going to start the same way that all of our complement pathways are going to start, and that's going to be with some sort of protein detecting the presence of a pathogen. And in the case of the lectin pathway, those proteins are going to be known as either MBL, which stands for mannose binding lectin, or we have another type of protein called a ficillin, and these are going to bind sugars on a pathogen's surface, so on surface. And this allows us to bring in two new terms, which we're going to see repeated throughout our discussion of the innate immune system. The first term is going to be what we use to describe the sugars on the surface. These are known as a PAMP, P-A-M-P. And what this stands for is pathogen associated molecular pattern. These are different molecules, different proteins, sugars, whatnot, that signal to the body that there is an invader, things that we would only see in non-host cells or in a non-host environment. These PAMPs are going to be detected by the class of proteins that MBL and ficillin belong to. These are known as PRRs, which stands for pattern recognition receptor. Let me make that R a little bit clearer. R. So we have a pathogen-associated molecular pattern, and these molecular patterns are detected by the pattern recognition receptor. So it's a pretty clear relationship. But let's actually dive in now to the PRRs of the lectin pathway. The first one is MBL. And MBL belongs to a class of proteins called a collectin. Specifically, this collectin has a C-type lectin domain. And we'll see what that looks like shortly. But let's discuss now, what does that C-type lectin domain bind? And what does MBL in general bind? And it binds to a few things. The first one should be obvious. Mannose binding lectin binds mannose. In addition, it binds two other sugars, one being fructose, and another being known as N-acetylglucosamine, which we can abbreviate as this, GLCNAC. So with that out of the way, let's actually show what this binding looks like by giving the structure of a single MBL subunit. It looks something like this, where this little circle thing on the bottom is the C-type lectin domain. If this is a pathogen's surface, then this would be the mannose on its surface, and the binding event would happen here. And it's important to note that this binding event is actually quite weak. We can say that this has a low affinity, that the MBL subunit, the single MBL subunit, has low affinity. This is quite weak. And this should be setting off alarm bells, because if we want to make sure that the body knows there's a threat, then we want to make sure that that signal is being propagated, and the signal can't be propagated if the binding event is weak. However, this is only one subunit, and as a matter of fact, MBL is made up of several subunits where you can have anywhere from 6 to even 18 C-type lectin domains included. So the real structure of MBL looks something like this, and we'll worry about these blue and green proteins in a moment. But what I want to draw our attention to is the many different subunits we have combined, meaning that when we have a pathogen surface that will be covered in these mannose sugars, in these other ligands for MBL, we have several different binding events happening at once, which leads to, in total, a very strong connection between the MBL and the pathogen surface. This summation of all of the binding events is known as avidity, and we can see that the avidity here is actually quite high. So we have high avidity, which means that the sum of all binding is quite high. And this is what we use to make up for a weak connection between a ligand and its receptor. Just add more binding subunits, which means that the overall binding strength is higher. With that out of the way, let's now move on to our second PRR of the lectin pathway, the ficcolins. And ficcolins, similarly to MBL, are a collectin, in this case with a fibrinogen-like domain. And what this leads to is it's going to bind different things. And moreover, ficcolins are a family of proteins. And the ficcolins that we're going to worry about, there are actually 3 types. The first is going to be known as M-ficcolin, also known as ficcolin 1. These bind acetylated sugars. The next class of ficcolins are known as L-ficcolins, also known as ficcolin 2. These bind something known as lipoteichoic acid. Lipoteichoic. I think I spelled that correctly. And the same sugar we saw with MBL, our N-acetylglucosamine. So GLC. The last type of ficcolin is known as an H-ficcolin. We also call it ficcolin 3. And ficcolin 3 binds D-fructose and galactose. So with our PRRs out of the way, let's now discuss the activation pathway. So remember, this is what our structure of either our MBL or our ficcolin looks like. Obviously, the domain at the bottom is going to change, but it's going to have a similar overall structure. And now we can bring in the proteins we saw on the side. This first protein in blue is known as MASP1, and the protein in green is known as MASP2. These are a type of protein called a serine protease, and these are going to be the things that will cut the complement proteins later down the pathway. But let's start at the very beginning, which is going to be that either our MBL or our ficillin, which we'll say is our PRR, it will bind its PAMP. And those are anything that we listed above, just based off of what the PRR is. But what this is going to lead to is a conformational change in MASP1. Which is just a fancy way for saying shape change in MASP1, our blue protein. And this is going to, in and of itself, lead to the activation of MASP2. And this is where things really start to go off, because we are going to introduce our first complement protein, C4. C4 will bind MASP2, which will lead to C4 getting cut. So C4 gets cut into C4A and C4B. And I left a little bit of space for a reason, because here is our C4. Looks something like this. And remember that our complement proteins always have a large subunit and a small subunit. And because C4A is not one of our exceptions, that means that C4A is going to be our small subunit, and C4B is going to be our large subunit. In our next step, we're going to have our next complement protein, C2. C2 will bind C4B, again, our large subunit, which will lead to MASP2 cutting C2. Into, specifically, C2A and C2B. And remember, C2 is our exception to the rule. So if we have C2, which I will draw on top, C2A is actually our big subunit, our large subunit, and C2B is our small one. C2B and C4A are going to dissociate away from the entire structure. And what this is going to leave us with is going to be what we discuss in step 4, which is what we're left with, that being C4B and C2A. This is what we call a C3 convertase. This is where every single complement pathway converges, the formation of a C3 convertase. And this will cut C3. So C3 convertase, which will cut, and I'll put it in purple, C3 into C3A and C3B. I'll draw it right below us, right here. We have our C2A. We have our C4B. These are now complexed together, and now C3 combined. C3A and C3B will be released when this gets cut. And C3A, remember, this is not one of our exceptions, is our small subunit. C3b is our large subunit. And these are going to be the primary effector proteins of the complement system. C3b is our opsonizer. Recall that an opsonizer is something that signals for phagocytosis. It binds to the surfaces of some sort of object, usually a pathogen, and signals for the object to be phagocytosed by another cell. And C3A has a similar function, but in a slightly different way. One of its functions is to be an inflammatory agent. It causes inflammation. And the second thing it does is related to phagocytosis. We can think of it almost as a dual authentication of the phagocytosis signal. So dual authentication of phagocytosis. Now, what do I mean by that? What I mean is that when a cell that can phagocytose a foreign invader detects the presence of C3b, it will begin to engulf that object, the foreign object. However, the phagocytosis won't actually begin until C3a binds to a separate receptor on that same cell. And it goes to make sure that whatever is being phagocytosed is meant to actually be phagocytosed. We don't want the host cells accidentally being eaten because C3b sticks to the surface. We want to make sure that the phagocytosis events are deliberate and meaningful, and so we need another signal to confirm that. And that's what C3a does. It makes sure that what we're actually devouring is what we want to devour. Moving on, let's now discuss the classical pathway. And the classical pathway has several things that make it unique. The first one being the pattern recognition receptor, the PRR, that it employs to begin its process. That being the complement protein C1. And C1 is different from the other complement proteins in that it's actually made of 3 parts instead of 2, and those 3 parts never separate from each other. Those 3 parts are C1q, C1r, and C1s. And if we're going to bring back the lectin pathway, draw comparisons to that, then C1q is like MBL or ficolin. C1r is like MASP1. And C1s is like MASP2. Moreover, the things that C1q binds is actually quite interesting. So C1q, again, is going to be the thing that binds to the PAMP, the thing that signifies that there is an infection. It binds 3 things. It binds lipotecoic acid, it binds an inflammatory marker called C-reactive protein, and it binds, and this is the interesting part, the constant region of antibodies. Why is this worth noting? Well, antibodies are generally associated with the adaptive immune system. Although there are nonspecific antibodies that are floating throughout the blood at any given time, we generally associate antibodies with, again, the adaptive immune system. So even though the complement cascade, the complement system, is generally considered an innate immune defense, the classical pathway kind of breaks that convention in that it only activates, or I should say, it activates most frequently after the innate immune response has happened or has first initiated. But let's actually talk about how this initiation works, and it will seem very familiar. So the first thing that happens is that, and I'll make the pen a little bit smaller, C1q binds its ligand. again, being one of these three: lipoteichoic acid, C-reactive protein, or an antibody. And when this happens, C1R activates C1s. An activated C1s can interact with C4. So C1s binds C4, which leads to C4 splitting into C4a, the small fragment, and C4b, the large fragment. This large fragment, C4b, can again interact with C2, and that's going to be our next step. C2 binds C4b, which leads to C2 getting cleaved, or I should say gets cleaved into C2a, which again, remember our exception, this is the large fragment, and C2b, which is our small fragment. And just as we saw in the lectin pathway, C4b and C2a combine to form our C3 convertase. And because the C3 convertase is made of C4b and C2a, it's sometimes known as the C4b2a C3 convertase. And this will again cut C3 into C3a, our small fragment, and C3b, our large fragment, and our opsonizer. The last complement pathway for activation is the alternative pathway. And the alternative pathway actually has two different routes associated with it. And by route, I mean way to make a functional C3 convertase. The first way that we can do this is to use already present C3b. And this might not make a lot of sense at first because we're using the products of a C3 convertase to make more C3 convertase. But this is actually something that's going to be really helpful for us, and I'm going to explain why after we go through the entire process. So the first step is that we're going to have C3b, and this is from either the lectin pathway or the classical pathway, and it's going to interact with a protein called factor B. And factor B is one of those complement proteins that we discussed in the nomenclature section that doesn't actually start with the letter C. We said that we were going to see these throughout the alternative pathway, and this is the first instance of that. So I'm going to draw what we discussed. This is C3b. Remember, it's our large fragment of C3, and it's going to interact with factor B. And factor B, because it is a complement protein, is going to have the same overall structure where we have two different parts of it that are connected and then later get cut. And how that gets cut is going to be what we discuss in the next few steps. So the next step that happens, and it's almost happening concurrently with step 1, I'm going to call it step 1A, is that we're going to have our second complement protein that doesn't start with the letter C called factor D. In this case, it's going to be pro-factor D. It's going to get cut, and in this case, that also means activated, by a protein called MASP MASP3. And yes, this is another MASP. We saw MASP1 and 2 in the lectin pathway, and in addition, we also actually see MASP3 associated with those same collectins that we saw in the lectin pathway. MASP3 can be found connected to either MBL or any of the ficilins we discussed. But if we draw it out here, here's pro-factor D. And then I'm going to show MASP3 just by these pair of scissors, because MASP3 is going to be what cuts pro-factor D into its active form. So this is MASP3. And now we can go on to our true step 2, which is that factor D, which is now active, cuts factor B into the two fragments BA, which is going to be our small fragment, and BB, which is going to be our large fragment. Just as we saw in the other pathways, so I'm going to draw the squiggle to represent factor D cutting factor B, the small fragment is going to go off. And this is going to now leave us with C3b, BB. It's a lot of B's, and it's also our C3 convertase. So when we first started to discuss this route of the alternative pathway, I mentioned that it might not necessarily make a lot of sense that we're using the product of a C3 convertase to make more C3 convertase. But this is actually really helpful to us, because that means that once the complement cascade begins, it will spiral and continue to grow exponentially, because if the product of a C3 convertase can make more C3 convertase to make more products of said C3 convertase, then we have a sort of self-amplification, perpetual increase in the activity of the complement system. So that's the first route. The second route is potentially even more unintuitive. So our second route is to utilize what's known as the tick over, and I'm going to put this in blue actually, tick over of C3. And what I'm referring to is the spontaneous cleavage of C3 into C3a and C3b. And this should be even more confusing, if anything, than the first route, because if we've been using C3 convertase to cut C3, and now I'm saying we have a way to cut C3 without it, then what's even the point of all the other pathways that we've discussed? And then in addition, if we have the products of C3, C3a, and C3b floating around, well, then what is there to stop the activation of the complement system, leading to destruction of healthy host cells? And to answer these questions as to why this isn't a problem, I'm going to need to draw in a little bit of organic chemistry. So I'm going to insert for us the active structure of C3b. When C3 gets cut, C3b is formed, and it exposes this red highlighted area. This is a thioester, and thioesters are particularly susceptible to acyl substitutions, meaning that if we have either an OH group or maybe like an NH2, a strong nucleophile with lone electrons to undergo nucleophilic attack, we can have the attack of the oxygen pushing the electrons up, back down, and then this will grab onto some random floating proton. And this is how C3b attaches itself to the surface of a microbe. This leads to the creation of this kind of structure. And now the C3b is adhered to the surface of the microbe. However, we can also, at the same time, if we just need a nucleophile strong enough to attack the carbonyl carbon, then we don't actually need a hydroxyl group from a surface. We could, just as realistically, have, well, randomly floating water do the same thing. It attacks the nucleophilic, or the carbonyl carbon, pushing electrons up and then back down, and then this will grab a proton. And what we're left with after a little bit of extra organic chemistry is just this form of C3b, where we just have a carboxylic acid group instead. This is known as fluid phase C3b, which we can also abbreviate as C3bH2O. In this phase, in this state, the C3b is essentially inactive. It can't adhere itself to the surfaces of any microbes nearby. However, this fluid phase C3b can still interact with factor B and then form the C3b-BB C3 convertase. And that's going to be what we see in this alternative pathway. We have the formation of fluid phase C3b, and this can then interact with factor B, which is then cut by factor D and MASP3. And what this leads to is this formula here. It's C3b H2O BB. And this is known as fluid phase C3 convertase. And this is a functional C3 convertase. It can convert C3 into C3a and C3b. However, all of that C3b that's made will be inactivated by this reaction, by the addition of water to, and the attack of that, the acyl substitution at that thioester. And so as long as there is no surface for the C3b to adhere to, it will be almost instantaneously inactivated by the water that is ever present within the blood. And so this doesn't become a problem. However, if there, instead of being the water that attacks the thioester, it is the surface of a microbe, then the C3b will adhere itself to the surface of the microbe, and then the opsonization pathway continues, which will eventually lead to phagocytosis. So the last thing that we need to discuss with the alternative pathway has to do with the stability of these C3 convertases. And the fact of the matter is that they're actually quite unstable. So both fluid phase and our normal C3 convertase that with C3b included is unstable. However, if we want to stabilize it, we can, and we can do it through a protein, and I'm going to put this in blue. It's going to be stabilized by a protein called properdin. And the properdin, we could think of it almost as if it wraps itself around the C3 convertases, the C3b-BB convertases, both in its fluid phase and in its normal phase. And this prolongs the half-life of the C3 convertase and allows it to continue to convert C3 even after its expected half-life. And this allows the alternative pathway to be a viable way to keep the complement system active. So that just about wraps up our first video on the complement system. We discussed the nomenclature used to describe the different proteins of the complement system and the 3 different pathways that exist to activate it. the lectin pathway, the classical pathway, and the alternative pathway. In our next video, we're going to discuss the further downstream effects and regulation of the complement cascade. But until then, this was Beyond 101.